Hello lovelies, in this video we're going to be looking at biodiversity for your A-level biology. So Dr. Edwards is going to take you through what it actually is and why it is so important.
Biodiversity. Biodiversity is the variety of living organisms in a given area or habitat. This can be measured on different scales. So a habitat could be something very small and very local, all the way up to something global. So a local example or a small scale example of a habitat could be a rock pool, a pond, or a single tree. This could scale up to quite a large ecosystem, such as a coral reef or a forest or a desert, or we can look at it on a global scale where we look at actual kind of biomes. So there's tropical forest in multiple places around the world, but tropical forest in itself is a habitat. Tundra is a habitat. Mountains are a habitat. Desert is a habitat. So we've got these kind of differences in scale, and we can measure biodiversity in each of these. It's important to still measure biodiversity on a local or a small scale, because even at a small scale, one rock pool, one pond, or a single tree could support really large numbers of species, plants and animals. It's also really important for us to measure global biodiversity, because then we can actually provide baselines in order to be able to know if that biodiversity is changing at any point, and therefore we can monitor the impact that, say, climate change or human impact is having on biodiversity in those areas. Having a high biodiversity is really important because it creates stable or balanced ecosystems. Something like a really diverse coral reef or a really diverse tropical rainforest, these are what we call stable or balanced ecosystems, because all of the species and all of their population numbers are stable and in balance. The environmental conditions are very stable and it keeps the population size pretty constant. So there's not big changes or fluctuations in the size of populations within this ecosystem. Normally this is due to large food webs, interconnected food webs, so there's a high interdependence there. So this means that small changes to the population size of one species wouldn't have a massive impact on the entire ecosystem, because there's lots of other species they can depend on for food, for example. Having high biodiversity, therefore, then makes you more resistant to change, whereas having low biodiversity in your ecosystem would be the opposite, because you would be more likely to suffer quite big impacts if one species was taken away or removed or migrated or went extinct. If a stable ecosystem is lost for some reason, so natural disaster or through human interaction, then it can take hundreds of years for that stable ecosystem to recover and get back to where it was. And the impacts on all the species that rely on that ecosystem is huge. So, for example, tropical rainforests can be lost because of deforestation, or coral reefs can be lost due to bleaching from high temperatures of the sea and also high carbon dioxide concentrations in the seawater where it dissolves out of the atmosphere. And those are obviously both the result of climate change. We need to know how to measure biodiversity. We've talked about genetic diversity before, and that is the number of different alleles in a population. It can be measured or at least estimated. It's quite hard, but you can use genetic techniques in order to do that. The other way of measuring biodiversity is looking at species diversity. This is otherwise known as species richness, which we can measure, and species richness is the number of different species in an area. Or we could measure the index of diversity. This is also known as species evenness, and it's actually potentially a better or more reliable measure of species diversity. This is where we need to measure the number of different species and the number of individuals of each of those species in an area. The index of diversity is considered a better measurement of species diversity because it takes into account both the number of different species and abundance of each of that species, so the number of individuals. This stops something like a measurement of 3 for species richness being misleading, because if in a field you've said the species richness is 3 because there's 3 different species, but actually it's like 99% grass and there's 1 dandelion and 2 daisies, that doesn't give you a real indication or a picture of how diverse that field is, because really it's just grass. Measuring biodiversity. Simpson's Index of Diversity is one way of being able to calculate and compare biodiversity. It takes into account both species richness and species evenness. You need to know the formula to measure Simpson's Index of Diversity. So it is D, or diversity, equals 1 minus the sum of small n divided by capital N squared. So small n is the total number of organisms of each species, and then capital N is the total number of all organisms that you sampled. And then remember that big E symbol means sum of, so you add them together. The result of this, so D is always between 0 and 1. The closer to 1 your index of diversity, the greater the diversity of that area. And obviously we want areas to be more diverse because that means they can cope with environmental change. We can also measure genetic diversity. Measuring genetic diversity is important because low genetic diversity in a population means it would be unable to adapt to changes in the environment. And this means it could be at risk. So being able to measure the genetic diversity of populations means that we can monitor this and maybe start helping to increase that genetic diversity. Populations that are at risk of having low genetic diversity are isolated populations or populations of animals in captivity. Polymorphic gene loci have 2 or more alleles. So remember, the locus of a gene is just where it is on a chromosome, or its location on a chromosome. So polymorphic genes are ones that have 2 or more alleles. For example, big A and little a, or big B and little b for one gene. We measure genetic diversity by calculating the proportion of polymorphic gene loci in a population. To do that, we take the number of polymorphic gene loci and divide it by the total number of gene loci. So this is the number of genes that have 2 or more alleles divided by the total number of genes. Let's have a look at an example then. I've got 2 fields, A and B. In A, my species richness is 2. I have 2 different species, a pink flowering species and a red flowering species. And in field B, I have a species richness of 3 because I also have a white flowering species. So my big N, my total number of all organisms for A is 36. And my little or lowercase n for my field A is 5 red, and then lowercase n for the other species is 31 pink. I'll do the same for field B. Okay, so here's our equation. Obviously, D equals 1 minus the sum of n, little n over big N squared. So let's work this out for each of our fields, and that will give us our Simpson's Index of Diversity, which we can then compare. So 1 minus 5 for the red species divided by 36, which is the big N squared, plus 31, which is the number of pink species over 36 squared. And we're going to add them together because remember that symbol means sum of. So we need to add those together. So that gives me 0.7608. I would literally put all of that, the bracket work into the calculator, not the 1 minus, but everything else into the calculator. Make sure you get your brackets done properly. And that's what it gave me. So then my 1 minus that number gives me 0.239. So that's my Simpson's Index of Diversity for field A. So I'm going to do exactly the same for field B. Just have to move my list of numbers over the way so I can fit them all in. But 11 divided by 37 squared, 12 divided by 37 squared, and then 14 divided by 37 squared. So I'll put my big N on the bottom, my little n's on the top squared, and then I'm going to add those 3 sums together as well. So putting that all into the calculator gets me 0.337. So I'm going to do 1 minus 0.337. And obviously you should be aware that because you're taking it away from 1, your calculated number that you get from all the brackets before you minus it from 1 should be obviously a decimal, a 0 point, less than 1 number, and then your answer is going to be less than 1, or it could be 1, but no more than 1. So then if we're comparing these 2 Simpson's index numbers, I've got 0.239 for field A and 0.663 for field B. Field B then is more diverse, or has a higher biodiversity because it has a higher Simpson's index than field A. That makes kind of more sense as well because there's more species, but also there's more individuals of each species. Because in field A, we only have a few individuals of the red species of plant, this is a problem. So if something small was to change in that environment, the red species could go extinct pretty easily. And that's why it's kind of a weaker structure if you have a lower biodiversity. Whereas with B, there's lots of each individual of each species, and they make up quite an even proportion. It's a more even, the species evenness is better. And so that makes it a more diverse and therefore a more stable environment.
Ouch! This is why in some videos I explain scratches.